Hello,
Hi,
how are
how's it
you?
going? Hi,
Oh,
Liz.
I'm
How's
good.
everything going? Good,
My
good.
clinic day managed to work around your session really well.
Yes. Awesome,
Worked out.
awesome. Hi, Julianne. How are you?
I'm great, thanks. How are you?
Doing well, doing well. Thank you. Thank you. It's a sunny
Good.
day where I am, so what's not to like?
Right, exactly.
So, Liz, which site do you operate out of? Like which of the more sites?
So today I have a clinic day. So I see patients from like 8 to 5 in the
Oh, wow.
hospital. And then Mondays I have a clinic that just moves around to different locations. And then Tuesday, Wednesday, and Thursday are basically my research days. And I'm at the experimental station at the lab, which is like a DuPont facility that's
Yeah.
like just across the street from the hospital in Delaware.
Nice, 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 nice.
Mhm.
Cool.
So I'm a nomad.
Understood.
Do you have one
Nice.
spot at DSU where you're located?
Yeah, yeah. So the university has like I think three campuses. And then there's another four sites. But
Oh,
I'm
wow.
I'm on the main campus. Yeah,
Okay.
I'm on the main campus. Yeah. So the different people do different things. So like there was a new acquisition that maybe about a couple years now. Another university in Dover. So there are a bunch of faculty that they're primarily based out there.
Okay.
But yeah,
Where's
I'm
the
in
main
Dover.
site? Is it in Dover?
In Dover, right, right,
Okay.
right, right.
Hello,
Hi,
hello.
Rob.
Hey, how are you,
Hi.
Hakeem? Nice to see you.
Good, good. How's it
Hey,
going? Nice to
Liz.
see you too.
Hey.
Yeah. I think we're waiting for Melissa, right? Harrington? Let's see if she sent any notes.
Oh, there she is.
Hey, Melissa.
Hey,
Hi,
Salt Creek.
Melissa.
How are you?
Good to see you all.
Nice to see
Good
you.
to see you. I feel like I've been seeing you a lot lately, Melissa.
Yeah, I'm lucky.
Mhm.
That way.
Nice, nice,
Nice to
nice.
see you.
Uh,
Julian, is there anybody? I think this is it, but I don't have my Outlook open to look. Who else is?
Let's see. It's Melissa. I think that's everyone. Yep,
Is
this is the
All
group.
right.
Yes.
Welcome. So I think, well, Liz and Hakeem, did you just meet? I heard you chatting about where Hakeem's
Oh,
lab is and everything. Yeah. You met
we've
before?
interacted a bunch before, but not for
Yeah.
like a longitudinal period of time.
Gotcha.
Right.
And have you met
Passings.
have you met Melissa Harrington?
No, I don't think I have.
So would you do a quick introduction, then Melissa, and then we'll launch right in. Bless
So
Bless
you.
you.
I'm
Bless
Liz
you.
Wright-Finn.
Bless you.
I'm a pediatric neurologist and I guess I would classify myself as a developmental neurobiologist at Nemours. And we study microglia biology in neonatal brain injury.
Great. Oh, nice to meet you. So I'm at Delaware State University. I guess I would have to say I used to be a neurophysiologist. Now I'm pretty much a research administrator, but gave up my lab in 2023. But still help out with and studied motor neuron development, and most recent the things that I still have a little bit of a hand in have to do with spinal muscular atrophy and um, pathophysiology in the brain. So we're moving up as they supposedly fix the problems in the spinal cord. We're moving up and looking
But
higher.
it's expressed in all the cells. So yeah.
Exactly.
Yeah.
So and
Mhm.
Melissa was also she was the PI on the COBRE, the Center for Neuroscience Research
yes.
COBRE.
Okay,
That
that's
was
right.
Delaware State and UD. And
It's
so yeah,
not
so
past tense yet, right?
well, it's in phase 3. So I should say
I know it's in phase 3.
it's in phase 3.
We're in year 2 of phase 3, but we're not quite in past tense.
I'm sorry, my mistake. So Hakeem, I think you know everybody here, and you've done these before, but just to level set for everyone, we always start off with the proponent, the person who wrote the proposal, kind of explaining what they want to do. And that's very useful, especially if what you wrote and kind of what we read doesn't quite match what you say. So just in a few minutes, you know, 5 or more or something like that. Can you explain the kind of the rationale for the study and what you're trying to accomplish with the proposal?
Sure. I first of all want to say thank
before you
you.
start,
Oh,
I'm sorry, Julianne, could you record this? Is it recording?
It's recording.
Okay. All right.
Yeah.
Sorry. Go.
no worries. So to start off, I want to say thank you to Rob and to all of you that are sitting on this panel. I'm so grateful. And Rob, when I do get an R1, not if, when, I want to join you on this to kind of help others as well. I've been looking for an
Absolutely.
opportunity like this for a long time. So it's nice to see it. I think I would have been more successful is what I tell myself if I had Mac R1 sessions like this before. So just jumping right into it. This is kind of my baby, this project, because it's what started my lab. When I left UCLA for the postdoc, I got a lot of like encouragement from the NIH. Is the nice way I would put it, to not do what my advisor was doing, not even go anywhere near that. So they put a lot of pressure on me to find something completely different. And so this was what I came up with. And I used this to apply for a K99. I got a 25. Very disappointed, didn't get funded. But then when I started at Dell State, I was able to get a KO1 out of this. And so this project represents the next iteration of this. I've been stuck at the transition to the R1 from this for the past few years. And so, Rob knows my story. After a while, I kind of gave up, but then I was encouraged to pick this back up. And so that's kind of the context of this. So the big question is to understand the role of changes in acetylcholinergic release in synaptic physiology and behavior during the lifespan. The backstop to this is, as we all know about Alzheimer's disease, debilitating. There are not many good drugs to treat Alzheimer's, and part of it is that we still don't understand the mechanism through which pathological aging like Alzheimer's arises. But it's even worse because even in normal aging, we still lack a lot of really high resolution understanding of how those processes unfold. So the purpose of this project is to shed light into exactly, how does it work with respect to the cholinergic signaling? How do they age? And then what has synaptic release of acetylcholine, specifically in the central nervous system, got to do with that process? And so it's on the basis of that I advanced this hypothesis, which has undergone a lot of changes over the years. But fundamentally, the idea is that there is a certain level of acetylcholine release that you need for normal aging, whether it's in the neuromuscular junction of the central nervous system. And then if you exceed that one way or the other, what I call a breakpoint, then all kind of bad things happen. It's very important to situate this in the context of dopaminergic signaling, for example, where release of the neurotransmitter is more concurrent with what happens postsynaptically. So if you release a lot of dopamine, you get a lot of synaptic release and a lot of potentiation of synaptic activity. That's not what happens with acetylcholine. In fact, acetylcholine appears to operate in what we call a set point model. There's a very careful balance we have to make. And so what this project is trying to do is to find that balance of acetylcholine release that will not kill the organism or harm it, but rather improve outcomes at points proximal and distal to the synaptic release site. So there are 3 specific aims that are really kind of geared to capture different aspects of that question. Aim 1 is synaptic physiology. So we just use electrophysiology to characterize different backgrounds of acetylcholine release. A lot of release, little release, somewhere in between. We've got some really cool tools in the lab that we can manipulate how much acetylcholine we're getting out there. And then aim 2 is the behavior aim. So one very important addition I've had to this since I submitted my last R1 is looking at sleep regulation. So we've got some really cool data in collaboration with our friends at UCLA that they've been trying to map out the cholinergic circuit during aging with us. So eventually I'll bring that to my lab, which is going to be part of the grant. But what we found out so far is that if you overexpress this transporter, you actually get the fly sleeping a lot more. And so we were very interested in finding out why, what circuits are controlling that, how does that get modified during aging? And then the other two are just cognition, which is pretty standard for these kinds of experiments. And then locomotion is another standard fair we do in the lab. And then finally is the characterization aim. So this, in a typical grant, this would be more like the slightly exploratory aspect of the grant, but here we're actually doing something more interesting, which is that we're trying to understand what defines those synapses in the first place. And then we're trying to also manipulate the synapses and then finding out how that affects the structure. So we're going to use electrophysiology. We're collecting some preliminary data on super resolution microscopy as well. So that whatever we find in aims 1 and 2, we can satisfactorily explain in aim 3. So I'll stop there.
Okay. Is my microphone on? Yes. So did anybody have questions for Hakeem? Because then we go through comments. Any questions for Hakeem?
I have a question. At what point does it become lethal to not have release of ACH? Like, is it pre-birth lethal?
Yeah, no, this is a great question. So it depends on what kind of manipulation you make to acetylcholine release. If you really completely shut it down, yes, they die just after neurogenesis. So the neurons do form, but then they don't make it out of the embryonic stage. If you have more like a hypomorphic state where you do have some, but it's not enough, you can actually get them to survive to adulthood, but then they die as young adults.
What do they die
or
of? Do you know what the
Great
actual
question.
cause of death?
Great question. We don't know that. And I tried to exploring that. The assays were not very good and they were raising more questions than answers. So I don't actually know, but my hypothesis will be that it's probably some metabolic effects in the gut because we saw some preliminary data to that effect, but I don't have the answer to that. I don't know why they died.
Okay.
Good question.
How strong is the background of, because as I'm reading through your intro, and I don't know if I'm jumping the gun, but I'm reading through the background information, I'm not hearing enough concrete facts to say this is really important. I have to study this. This totally flows. So I want to know, how strong is the evidence for this acetylcholine release and or concentration in the synaptic in the synapse,
Right, the cleft.
essentially.
Yeah, no, that's a great, yeah.
And
Sorry, go ahead.
can we be sure that it's involved in the aging process, or is it just that it's changed in the aging process and it's not actually causing aging? It's like a representation of something that does occur during aging naturally.
Yeah, that's an excellent question. And the answer is that it's very murky. That's the bottom line. And that's actually what helped me get a job 12 years ago, because I was very surprised too. I thought there would be a lot more concrete cause and effect, but it turns out that much of it is exactly what you're saying, which is that, yeah, aging occurs and then cholinergic system is messed up. Okay, maybe that happens with dopamine too, or serotonin, right? What makes the cholinergic system different is that you need it for cognitive view, right? You need it for sharpness. You need it for a bunch of stuff. At the NMJ, you need it for your muscles. So that's really what kind of raises the bar. But the evidence is not very concrete. It's a lot of anecdotal. It's a lot of indirect. Much of it was done in humans, which there's nothing wrong with that, but it's very correlative, some of our experiments are not the first, but there are some of the few out there that have systematically gone after that question of where is the cause and where is the effect on this. But it's a great question. It does mean I have a lot more to do in my background and significant section, because it's a good
Well, and
point.
you're alluding to you're seeing behavioral changes or maybe sleep-wake disturbances in your manipulation of this that I feel like belongs somewhere in there to lead us down this path,
Oh,
that
thank
there
you.
are consequences of changing
Oh,
the
thank you.
system. Because I think that's an important point for us to know, is that you've already
Thank you.
done
Thank you.
this and that did something. And so that tells us
Oh,
that doing more things and really investigating what that did is going to tell us more about how the system works.
thank you. Thank you. I'm writing that down. Thank you. Thank you. That's good.
So you want to keep going,
Yeah,
Liz?
I had a similar, just to piggyback on that, because I had a similar concern. I thought, because I'm more familiar than Elizabeth is, I'm more familiar with the work you've done because I've seen your student stuff, presentations and everything. So I know you have a lot of background information in there, or a lot of background work that you've done, preliminary work, and you don't mention it at all, except like one line in the middle of AIM 2. And so you need a much clearer, in those first paragraphs, a much clearer kind of laying out the research path that led you to your hypothesis. And I think what you've used up your space by really going into not quite detail, but the 100-foot level of what your experiments
Right.
are going to be. But what you need to do is turn that into the 1,000-foot level so that it takes up this much space instead of half the page, that it takes up this much space so that you can have more space to go through that work that Elizabeth was saying is going to help us understand how you got there and lay the groundwork for it.
And I agree with that too. And I would add, I think there's a lot in here that, and I'm familiar with your work too, but just taking a step back and looking at it, the sequence that you present things in, if you took more of an approach to sort of that 1,000-foot view, or sort of broad, and then focus down to very specifically, it would help reviewers, and Liz can comment on this because she might have seen this for the first time, follow. So when by the time you get to the first aim, or even the central hypothesis, you've already led them to it. Because the sequence now, it's almost like you talk about the transporter, but that just sort of comes up. My brain, like, objectively, I wasn't led to think, oh, the transporter is going to be important here. And so part of, I think, my suggestion is just think about, well, how do you take somebody, a reviewer, who will know a lot about sort of the fundamental neurobiology, maybe some about the organ, like the system you're going to use in the organism, but not really where your research question is coming from, and just start really big. What's the big problem? What are you addressing? What are you carving out here? Sort of what led you to think this, which is your results, which are kind of buried down below. And I agree with that, because that would allow, what you're trying to do is get the reviewer to almost think of the aim before you tell them what the aim is, right? So they're on board with you from the very beginning.
Yeah, like
Yeah,
by the
and
time you get to that
vast-
hypothesis, it should be like really obvious that that's the hypothesis. So you're like leading them down the little like logical pieces, like really obvious, so that by the time I get to your hypothesis, I'm like, oh, well, that makes
Of
perfect
course,
sense.
right?
Sorry, Melissa.
And no, that's all right. No, it's good. And just to point, like the thing that you're really studying, going back to how you introduced it and how that compares to what you wrote, the thing you're really studying is acetylcholine in the synapse, right? Release of acetylcholine. The VAST is the tool to manipulate it, right? So, but because it can kind of get bogged down a little bit into, oh, you're studying VAST. Well, not really. Right? The VAST is the tool to manipulate acetylcholine synaptic function.
And so in aim 1, is your aim to actually find out if your system works, or do you know that your system works and you're actually testing a different question?
Yeah, no, that's a good question. Yeah, we know the system works, but the extent to which it works, we don't really know.
Oh, okay. So it produces like a behavioral change, but you don't know if it actually changes the level of acetylcholine.
Yeah, and for that matter, like the synaptic physiology of it. So how is this? How does presynaptic release play into that, right? So because there are many ways that you can imagine it working. And so these physiology experiments will really help you to look at the levels of a presynaptic release, the excitability of the neurons. So all of these very important details will kind of really point you forward. I mean, to your bigger points earlier. Yeah, so that would really kind of help you there. But we know that the system works, we just don't know how well it does.
Mhm.
You step back one, so from that explanation, could you back to the central hypothesis? Because I think even the fact that you had to put the word breakpoint in quotes, that I find that a little confusing about that there's a breakpoint. So do you mean there's a threshold? Do you mean there's sort of a inverted V response? You know, there's I
Right.
think too much ability to interpret that
I
breakpoint.
see.
Or maybe
Okay.
that it's like a
Thank you.
tunable
It's
system,
It's
like
tunable,
there's
right?
modulatability kind of a thing of the system.
Yeah, no, great question. Great question. So yeah, breakpoint, I can see as soon as you said that, Rob, I totally see that. So yeah, what I mean is that there is a I think you use the exact word. It's actually an inverted it's like a V. So there's depending on whether you're looking at, if you have too little, you have a dysregulation. If you have too much, you come back to the same place. But if you have the right amount, you have the optimal effects. So yeah, breakpoint actually doesn't do it justice because it doesn't have the 1 point. But yes, yeah.
So is there,
So
is it more like there's an optimal acetylcholine release profile that's necessary
correct.
for functional synaptics? I mean, that's sort of what I thought you meant, but you don't want reviewers.
I'm going to steal that.
You
Yeah.
can steal that, yeah. And that allows you, because you don't have to, I mean, you have to, the central hypothesis has to be succinct. You want it something that'll stick in their head so they understand why you're doing everything else.
Right. So Rob, you said optimal something. I missed the other part.
Acetylcholine release profile.
Yes.
Which is still a little fuzzy, right? But
right.
I think it's
I'll work
closer,
on that.
right?
Yeah. Right.
And so still staying on the central hypothesis, that optimization changes over time. The optimal release, this is where I record, Aaron says, changes over time.
Yes, exactly.
Just for the sake of, reviewers, devil's advocate, acetylcholine gets like esterified, right, in the synaptic space, and then it gets broken down to become deactivated. Are there
Right.
other mechanisms that you could use to test the same thing that you could also maybe use? Is it easier to do this or better to do this for some reason? Like, are there other ways of tuning the system, or do you need to consider that you may need to, address these other pieces to really optimally tune your system?
Excellent point. The answer is a solid yes. In fact, going back to Alzheimer's, the treatments are cholinesterase inhibitors, right? Not all of them. There are others now, right? The treatment, exactly. The treatment. So yeah, those treatments, they don't like, back to Melissa's point about how VASH is a tool. So those do not manipulate acetylcholine at the level of release. They manipulate, like you said, postsynaptically at the synaptic cleft to stabilize acetylcholine. So if you inhibit the acetylcholinesterase, you stabilize acetylcholine. In theory, you bind, postsynaptically either with the ionic ionotropic receptors or the muscarinic, blah blah blah. So yeah, the answer is a solid yes. And thank you for saying that, because I have to say it was not in my original grant, and I think you're absolutely correct that a plan B at a minimum in the analysis section would be
right. Because if it's a system that can sort of reset itself back to normal, as many neurological processes can, due to shutting down of the receptors, then it may be that overexpression in the long run does change back to its set point somehow. And I don't know if that's true or not. You may
Excellent.
already know that.
Excellent.
Or maybe in the case of disease, it can't do that. So I'm just wondering if you already know stuff, then you may be able to modify what's in AIM-1.
Well,
No, this
yeah,
is a great, thank you. Thank you.
as the molecular cellular person, though, I would maybe it can add an extra nuance if you're really going to put it back there, because so acetylcholinesterase impacts only the postsynaptic side. But if you're overexpressing acetylcholine and charging up vesicle filling, vesicles fill faster. Maybe that stimulates more vesicles, right? There are a whole set of things that probably happen on the presynaptic side if you're greatly accelerating how fast vesicles fill or dramatically slowing how fast they fill. So I think acetylcholinesterase isn't just another way to, right? It's going to be
It's a whole different way.
In some ways, it could be really useful to compare, but you wouldn't want to suggest in any way that those are equivalent things. Doing those two things might somehow be equivalent. Yeah.
Okay.
Right. No, I agree. Sorry, Rob.
No, I have a curious question, because mostly my understanding of synapses is NMJs. So are there any retrograde, and this probably would be more in the background and significance in describing the circuit and why this loading part is so important, because the idea is you're getting a response. But are there retrograde signals back from the downstream neuron?
Yeah, I don't know. And in fact, combining what Melissa and Liz mentioned and what you said just now, that's why I like Liz's idea, because I don't really know that there are retrograde signals, but I don't know that there would not be some, right? Because some of the data that there's a new postdoc in the lab that we're starting to collect is really aimed at going after that, because there's no reason to think that, back to your point, Liz, that there's not some system presynaptically that would correct for that increased activity that you cause, right? And in fact, we see some of our overexpressors that they actually kind of begin to have compensatory effects as a result of that. So I think at a minimum, writing that into the grant would be very important. The reviewer will say, oh, this guy knows what he's talking about, blah, blah, blah. But more importantly, that there is something else that you're actually trying to get the full picture. So to your direct question, Rob, I don't know the answer to that question. That is,
I wouldn't sorry, Hakeem, I wouldn't suggest going after the whole thing in this grant, but at least staging it so that, and again, I don't know for sure, but the reviewers will, and maybe Melissa knows. It's the circuit that matters. And if you're looking on the presynaptic side, and there are no retrograde or known retrograde signals, you can just acknowledge that in the writing. If there are, you have to say, well, downstream, additional experiments, sort of future experiments, you can integrate that information in, and then try to focus back on the presynaptic pathways that you really want to look at.
Are there choline transporters on the presynaptic side?
Yes, there are. Yes, there are. In fact, Randy Blakely, one of our friends, he does a lot on that, and he does high affinity choline transporters. So there are. And then also, actually, in addition to that, oh yeah, so I was gonna say, like, there's a reason why, Liz, you were mentioning, cholinesterase inhibitors and there's a reason why, right? And I think part of the reason is because, Melissa, like you said, you're interfering at that stage, like it's very late to be acting, and whatever it is that you're doing is very obviously going to have a lot of side effects, which is what you see with those acetylcholinesterase inhibitor. Having said that, for a long time, that's all people had, right? It's causing a lot of stuff. You have side effects, you're vomiting, there's muscular issues, but that was what people had. It was doing something. So acknowledging that, like Rob said, without necessarily just painting a target on the back,
Mhm.
is a really good idea. I think I'll write that down actually.
No, I think you can use that. These are, chemicals used for the treatment of cognitive decline in human diseases like Alzheimer's, blah, 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 as evidence that this is an important part of the way that the brain works for cognition and certain behaviors. Whether my point was mostly just that there is no treatment for these diseases in reality. I mean, they're ultimately degenerative diseases that we sort of know how they work, but we don't really actually have a treatment for them. We just have a way of making you less bad early on.
Yes. Yes.
But that's, I think, good
Very
evidence.
limited. Yeah.
Yeah,
very
I mean,
limited.
if you
Make
catch
it-
it at the right time and treat it at the right time, maybe in certain people, this does help.
Right.
And so
Right.
that is, I think, good evidence to suggest that this does work well on cognition and memory. So,
So
yeah.
you have a cool diagram, right? I think I've seen you present a diagram
Yes,
with
I
the
do.
transporter and the whole. I think including that again, you'd have to do this relatively quickly in the grant, but to lay out, look, the circuit, right? That's the problem here. The treatments
Right.
address the cholinesterase, and that's really just a stopgap,
Yeah.
right? And what you're addressing is a critical unknown, right? This
Right.
is where the
Right.
hole is. We don't really understand this presynaptic part, which is critical in the pathophysiology of the disease and leads to future treatments. That would help with the significance and kind of getting the reviewers to understand why you're going after these specific presynaptic modalities, right? These specific, using the transporter as a tool becomes obvious because of course you need to use that as a tool. That's where you're looking.
Mhm.
Yeah.
Yeah, so that
Sure.
maybe and the to start the significance section might be a really good way to
Yeah.
start off the significance section.
Yeah.
And there may be alternate uses for these medicines as well, or this type of technology, trying to tune a synapse. Like, that's not just neurodegenerative disease. I don't know that it's ever been studied in things like this, but like in recovery from brain injuries, like traumatic brain injuries, we often have problems with like sleep-wake cycle that impacts their ability to engage with therapies, and so helping them stay awake and alert. Impaired, you know, learning and memory after various brain insults. Maybe developmentally, it could be harnessed for other things. So I don't know that there's any evidence for any of those things, but those are possible
Right.
ways of
Right.
tuning a cognitive
Right.
system
Right.
that can maybe
Yeah.
be more broadly applied too.
Yeah. Yeah. Great question. Great point. Great point. Thank you. Yeah.
And a comment that I had that I put in, but that sort of relates to that when you're talking about why study this in Drosophila, making the case, something that I think I think you should have in your specific games is the information that when you're disturbing acetylcholine transmission in flies, you're not touching the neuromuscular junction, to make sure people understand that. And that's why Drosophila is the perfect model organism to study central cholinergic processing and look at the impact on behavior, because you're not just paralyzing them.
Yes.
Right?
That is a
So,
really good point.
Yeah,
I agree
that's a
with
good
that on your
point.
specific games
I think
page.
that needs to be in the specific games in the top half before you get to the actual aims, to
Okay.
make that
Okay.
case that it's
Okay.
because people can be skeptical that Alzheimer's and flies could be that related, or these other neurodegenerative diseases you're talking about, that flies
Right.
really would
Right.
make a
Right.
good model to study that. So
Yeah.
Mhm.
you make that case that flies are uniquely good as a model system for studying cholinergic central signaling. I think that'll help.
Mm-hmm. That's a great idea.
Mhm.
As an aside, since this sort of comes right after what Melissa said, do you know what study section you're going to send? Like, right, you can suggest study sections and Have you looked to see, is there a fly-friendly?
Not, not,
Not yet.
yeah, not yet. So yeah, so but I did talk to the program officer, and I'll be reaching out to them again to suggest some. But I haven't done the due diligence on that just yet. So I'm going to try to go and look at the rosters and
Yeah, there's
yeah,
got to be
because that
one.
will be
this
really
is-
important.
It's an important for these at risk. It's important consideration because at risk these R01s go to a normal study section. If you wanted to do
Yes, they
the
do.
reward one for the DEI, those are being pulled into a special.
Yeah, and then they'll pull,
special
yeah, they'll
study
pull
section.
reviewers that match, kind of thing.
Yeah, but
Yeah.
but for this mechanism,
Yeah.
you can pick your
Yeah.
study section.
Thank you for bringing that up, Rob.
You're welcome.
Mm-hmm.
Yeah, you
So-
and the PO will get it, but you want a fly-friendly neurobiology sort of. You want the
Yeah.
right people, so they don't spend the reviewers don't spend a lot of time looking stuff up and trying to understand what you're doing.
The other thing to be cautious about, so I went and looked about how many of these have been funded. So since 2022, so they funded 16 in FY23, so which they've only funded 16 of them. And none of them are really molecular. None of them by the title looked molecular cellular. So just
okay.
so
Yeah.
I
see.
it's not like these are easy. My guess
Right.
is that they're kind of program officers might be using this as a mechanism for funding R01s that get a score of 35, or 30 in regular study sections. So
What's the PAR? What's what's the? Do you remember? But
no, I find it. Yeah.
okay.
I had looked at it, but
so
I
had it
can't find
open
it
last night. Because I looked it up on Reporter by the PAR.
It's PAR 22-181.
22-181.
Yep, that's the most recent.
There we go. Nope.
Mm.
Yep.
Yeah, it made me think
So
that
Lisa,
you could try. So it's good that you're trying this, and then but you might
Yeah.
if it doesn't if you don't get if it doesn't work out very well, you might want to check so that the reward ones have not been there's not been a cycle of awards yet. So we don't know what that's going to look like. But down the road, you might
So
it might be friendlier in that special study
Mhm.
section that just
Yeah.
looks.
So Melissa, I'm actually doing a reward for a different for the Parkinson's
Okay.
project is a reward. Yeah, that one I'm
Okay.
super excited about.
Oh,
but
great.
yeah,
So
for
you'll
the
have 2 data points.
yeah.
There you go.
Yeah. So for the at-risk one, this is my best shot at it just because I have the most papers on this one. I have solid collaborators, very good data, but I'm not disillusioned. I know it's going to be very tough. I mean, what the program officer told me was that she said that she can tell me that the pool is smaller. That's what she said. Because I asked her the same thing that how many people am I competing against exactly? She said,
Well, and the other thing that concerned me, okay, so when I looked at just the awardee, those 16 awardees, Stanford, Emory, Wake Forest, they're funding and people with Hispanic surnames at
Right.
really high at medical, and then there are a couple of medical centers. These are not going to small institutions, right? They're not funding.
Is that the
Good
DEI
people
or
in-
this mechanism?
Yeah.
No, this at risk. They're not funding investigators at small
Yeah.
institutions.
What's-
They're funding minorities
Yeah.
at major
yeah.
research universities.
So do we
Right,
know the
Stanford and
success
Princeton.
rate for this versus a traditional R01?
Well,
It's pretty new.
so we don't
I
know how many.
Yeah.
We don't know what the pool they
Yeah.
got. I don't know what that's the unknown, right? What was the pool they got?
Right.
They probably won't
Yeah.
release those data until they have enough people You know,
Yeah.
and I don't, you can ask I just looked in the PR because I was trying to find out who the NINDS program officer is, but they don't list one. It's just it's the GMO that you have to talk to
Right.
for
Yes.
NINDS. But just because they have been funding people from those institutions might just reflect those institutions responded quickly and got grants in.
Just
The
Yep.
intention might be to
Yeah.
go for
Yeah.
faculty. Yeah. So don't be too
Yeah.
discouraged. You might be
No,
the one
right.
they're
I don't
looking
mean
for.
to.
Yeah. Yeah.
I don't mean to discourage you. I just don't mind to say to eyes open, right? Eyes
Mhm.
open, and also the importance of the
Right.
kinds of things that you're doing. Yeah, you got to make a really tight case
Right.
that.
Yeah.
Which reminds me, speaking about making that case, the other thing I thought was missing is your team, your research team. That needs to be in the specific games. You need to talk
Oh,
about the
okay. Okay.
qualifications, and your environment needs to be part of that too.
Mhm.
Okay.
Um,
This is good. Okay.
because electrophysiology, right? You have the core supporting you. You have a
Yes.
core supporting you. You
Right.
have
Right. Right.
tremendous expertise in
Right.
Zhongli,
Right.
and so
Right.
you want to
Okay.
say a little something about that
Yeah.
on your
Okay.
specific game
Yeah.
stage.
Yeah, there's a lot to get in there,
Right.
but
So
Mhm.
Melissa.
it is important. Mhm.
Yeah. Like real estate. Yeah. So Melissa, do you think that like the fact that it's like for like new and special and ESIs, do you think that would at least like reduce the competition in a way? Like, because if I was doing a standard R1, I'd be competing with everybody else, not just folks at, you know what I mean?
Well, I think, yeah, because it's given that they're sending them out to whatever study section they apply to, you're competing for a separate pool of funds, right? I think
I
because that's the
see.
way they
I
do it
see.
at NSF with RUIs and these
Right.
ERIs, like that's the way they do it. It's all reviewed one, and then the most competitive of the things that qualify, for this particular mechanism
Right.
are then going to
Come
get
out of
pulled
it.
out for a separate
Right.
pot of money.
Right.
Yeah. So the advantage you have is that I think what you're going to need is just a reasonable score, a good score, and then
I see. I see.
the percentile isn't going to matter. Right. I think
Right.
I
that's
see.
what's going to,
I
because
see.
you'll be
Got it.
being pulled into a
Pulled
different
into a
funding
different
pool.
pool. Right.
Yeah.
Okay. Got it. Got
That's my guess. No, I don't know for certain, but I'll bet
I
how
see.
they're working it.
Okay. Okay. So,
Yeah, and so that's, I think, why what your program officer was saying when she said the competitive pool is smaller. She's talking about once you have a score,
Mhm.
then yeah, those percentiles are not going to. You're going to be compared against other people that are in that same applied to that mechanism, and
got it. Okay.
yeah, so
Excellent. Liz, I had a question for you as well. So you had talked about that question about, I think you had talked about like the lethality. Like, so to what extent should I be thinking about that? Because I don't have an answer to that, but then I don't want it to be something that somebody's going to get stuck on, right? So like with you, like having just, recently kind of just started looking at this, like how much would it bother you, basically? Right, that
So I think you can use it as part of your argument for why this sort of tuning system is a better way of getting at how this receptor sort of synapse works. Because if it's lethal from a very early age in all organisms, you can't study it. So you have to come at it from a different standpoint, and you can say, this is a critical piece of, normal biology across many species that's conserved. And so we can't, I mean, obviously you wouldn't say it this way, but essentially that you can't completely eliminate this receptor because it's critical for life. But you can tune the system using your method. And so it's a unique method to modulate a critical signaling mechanism within the brain and body. And then I think that leads you into, and furthermore, Drosophila is the perfect model species to do this, because we can specifically modify the central signaling without causing lethality from body effects. Whereas in animals, like you're saying, in the gut, there's a ton of acetylcholine and you can't transit probably. There's a million ways to die from gut transit problems.
Yes, yes, that's true.
So
That is true.
I think
That's
that that is just part of your argument that sets us up to realize this is really important, why we should care, that it's necessary for survival, that it's
Right.
dysregulated in aging and memory impairment. And that's why we have
Correct.
medicines that specifically target this system for humans who have cognitive problems. You want to better understand how it works, and you have the perfect system to do it. And then these might then cause us to better understand a system that we can design new therapies for any number of things.
Yeah, I wouldn't make a big deal about the lethality because it's an important part of the system. So I think that's sort of, yeah, it's like Liz was saying, it's because the system won't function without it. Of course, if it's null, they're lethal.
Right.
It's going to be lethal. So I don't.
Right.
That's something I said. I think I would just take out lethality altogether. I don't think, especially in the specific
Okay.
games page where
Yeah.
your
You
spaces
can
Okay.
put it someplace else.
put it
Okay,
someplace
okay.
else. Yeah.
Okay,
And focus
okay.
on
Okay.
the value of being able to move the system up and down. Yeah.
Down. Got it. Okay, thank you.
Because you're not
thank you.
studying what happens in the absence of VAST, right? What's going to
No,
happen
no.
is the animal's going to die.
That's easy. Okay.
So it doesn't need to be part of the specific aims.
Right.
So
You
and then I have some specific, I have some comments about specifically what you could take out to make room for. One thing is, even in the specific games page, I got lost in sub-aim 1, sub-aim 2, sub-aim, all these all these sub-aims and all that. I think that's, I mean, you got to go to the 1,000-foot level where you just say, we're going to look at 3 things. As part of the same, we're going to look at 3 things, right? And put it all in one sentence,
I
these
see.
3 things, because when they want the details, they've got the
They'll
research
go to the
strategy
research
section.
plan.
Yeah. And then you'll get a lot more room. Plus,
Right.
not giving a lot of details that somebody could get stuck in their mind on that
Right.
specific games page before they've had enough background to really digest it and wind up, with a prejudice that's going to keep them
Right.
from really appreciating
Right. The rest of
the way
it.
you use it later. Yeah.
Yeah. I think like probably most of that page should be background in order to really
Excellent.
lead
Okay.
us in, because like maybe I would say your aims take up like 65% of your page, just eyeballing it. And so if you
I
flip
see.
it
I
and
see.
65%
I
of your
see.
page
I
is
see.
background, then we're going to know
You'll
what's
be-
going on by the time you get down to the aims. You can be a lot more vague in those aims and then put like an impact statement at the end where like,
I see.
because of these experiments, then like, or these experiments will allow us to blah, blah, blah. This is the overall impact. So by the time they're done, they're like, okay, this makes a lot of sense.
Yeah. Yeah. Your impact statement, you don't really have much of an impact statement. Something else that you could consider for the aims, I think definitely you want in your summary, because the summaries, I didn't go through all 16 of them, but I went through about 5 or 6 yesterday looking at the things that have been funded from this. And 4 or 5 of the 6 I looked at, their last sentence was about career development, was about how this grant, this program was going to help their career development. So it looks so I would put that in the summary at least. And maybe think
Okay.
about it as part of the specific aims to remind reviewers what they're reading. Right,
Mhm.
that's not an ordinary R1.
Mhm.
Yeah.
Mm,
Yeah.
Yeah, it's a great
Yeah.
point, especially since it's going to go
it's
to
a
a
great
standing study section that's going to have other,
point.
straight up
Right.
R1s to look at.
Right, right. Oh, thank you. That's very good. So last sentence after the impact statement. Yeah, this is a good point.
Can I ask you to explain a little bit more about, I know this is a draft, but specific aim 3, what
Mm.
happens in aging, right, neurons and synapses? So what's the, I got a little bit lost there.
Okay,
Well,
sure.
and I built a lot into this because I'd heard you speak before and everything, but that one I got a little lost on what's the specificity in that aim.
Right. No, sure. So this aim essentially just seeks to characterize really those synapses in aging versus young flies. So what's actually happening within the cholinergic neurons themselves, and then at the synaptic botons where they connect with postsynaptic sites. And to use, I think I was planning to do ultrastructure, so EM counts the synaptic vesicles, and then we're going to actually stain for vesicular acetylcholine transporter. So we use immuno-EM, basically. We will be counting synaptic vesicles in cholinergic neurons. And then, yeah, so it's a heavy characterization of the synapses themselves. How are they doing? How many, ideally, and I talked to Jeff Kaplan about this, and we might be able to assess the synaptic, the number or at least the density of transporter on the synaptic vesicles. He thinks we can do it, but not sure yet. We're going to do some preliminary experiments in a few weeks. So, but long story short, it's just a characterization of those cholinergic synapses in young versus old flies. Are we distorting the morphology in any way? Are we altering the structure? To probably or possibly explain some of the phenotypes that we already see, right? So that's the.
So could you flip it? I mean, I get it better now, but can you flip that around so it's hypothesis driven? Because I think
Okay.
the way you sort of set it up, and even the way you describe, they're going to say this is descriptive, which is actually needed. It always drives me nuts when I'm in study sections and, well, this is just descriptive, or they're just characterizing the system. I said, well, that's actually very important to do. But I think you'd be better off maybe going after, what are the ultrastructural effects of, and then stating a hypothesis that you're testing.
Very good.
Yeah. So you end up doing the same thing. You just pitch it to the reviewers in a way that you're testing hypothesis.
So as an alternative,
you have some expectation. Go ahead.
Yeah.
Sorry, Liz, go ahead.
No, that's okay. As another way of approaching it, my thought was it feels maybe like part of aim 1, where you're really seeing if by altering the overexpression or expression differences in that synapse, if you're changing the structural makeup of that synapse. And so you might well be changing it because you've totally changed the system and the burden of the signaling through that synapse.
Right.
And so you could incorporate it into aim 1 potentially and say, when we overexpress this receptor or this, release of acetylcholine, then are the effects that we're seeing downstream due to a change in the structural components of the synapse, if I'm understanding you correctly. So you could also say, as a means of understanding the way that we've modified the system, we're going to look ultrastructurally and via whatever methods you're going to use to confirm that that's not a change to the physical makeup of the synapse that's causing the end result in the behavioral differences.
I like that idea. I mean, you could keep separate the functional / physiologic, sort of what you have in aim 1 now, then go to structural, ultrastructural, again, hypothesis driven is aim 2, and then
hypothesis driven.
yeah,
Yep.
and then aim 3 could be behavior.
Okay, I like that. It would be the
Mhm, mhm.
behavior.
Mhm.
Oh yeah, that's great. Okay,
Yep.
okay.
And
Does that
then
tie-
also,
that-
I feel like it should have a title to the aim rather than a question.
Okay.
Like what is it that you're doing rather than what is the, like, we
Right.
don't understand how it works kind of a question.
Okay. Okay. Got it.
Mhm.
Got it.
and each one should have a hypothesis for the reviewers to hang their hat on, so they so you don't get those comments about how it's just descriptive, right?
Mm.
And it really is, the way you talk about it, it really is testing hypotheses. You just have to present it that way
Mhm.
with an expectation. Like Melissa said, I think that's important. You're expecting this change over time, or this change with altered, when you alter the function, that this is what you're expecting to see.
Oh, this is great. Okay. Pretty good, yeah. Okay.
Right, and it might leave, by the time you have an hypothesis for everyone, that might leave only one sentence for you to describe
Correct.
what the experiments actually will be, but that's okay.
That is okay. Yeah.
Okay.
You just have
As
to,
long as-
it's a balance. You just have to give them enough to understand and to go back to. So after they read the whole thing, they kind of have it in their head. The Ames pages, the way I use it is just to reorient me. Oh, this is the one I'm talking about now, right? In a stack of all these grants. But there just has to be enough information in there to get them on board, so they understand what you're going to do, why it's important, and that,
Right.
your group is the one to do it. That's what you want the Ames page to sort of say.
Mhm.
Yeah, the hard part is making it one page. That really is the trick.
Yeah, it's like a poem, right? Every word
word
has a purpose. Every word has a purpose.
Do you have?
Do we want
Go ahead.
to talk just for a minute about the biosketch? I just had a couple
Oh, I
of,
forgot about that. Yeah, sure.
just a couple things that I want to say, which is really pretty small. It's good. The thing that there's a typo in the second sentence because you have the same abbreviation, AD, for Parkinson's and Alzheimer's. So you want to fix that.
Okay, sorry, I'll fix that.
The other thing I would say is you're leaving some things on the table by only having 3 contributions to science, and that means that there's 2 of your publications that you can't use. Actually, technically 3, because at one of your contributions you have 5 things, and we're only allowed to have 4. So you should tweak your contributions so you can add another contribution, because that gives you 4 more slots for publications. And
Okay.
I would put them in a different order. For me, I think you wanna, people pay the most attention to the top, and their attention flags as they go down. So your most recent, so right now you have it kind of chronological. Flip it so that the first contribution has your most recent publications, and put the oldest publications in the last one. Right, so
I would
that
say,
would be my advice.
I don't remember what your order of your contributions are to. I would start with the newest contribution. A lot of people start early in their career and they go later. Same rationale. The
Right.
contribution that's most recent and most relevant.
Most recent, most relevant. Yep.
Yeah, put that one
Mhm.
first, and then as they're tailing off, they can see all the cool stuff you did earlier in your career. Does the biosketch directions? Is there guidance on the order? I'm just
I
unless
don't
No,
know.
I don't think so.
Yeah, no. Okay, yeah, because I always thought the order was from oldest to newest, but yeah, it's true. You're
You can
right.
do
It
it
makes
any
more
order you
sense.
want.
Yeah.
You can do it
Okay,
any order
okay,
you
that's a
want.
game
Yeah.
changer.
I mean, that's how your CV is, right? That's the expectation in your
Right.
CV is you go old to new, and people look at the bottom.
Right.
That's a good idea.
Thank you.
And
That's
that
all.
was it. That was the only things. I just wanted to say those 2 things.
Yeah.
Thank you.
So do you have any other questions for us, Hakeem? We only have a couple minutes
No,
left.
no, this is all great, great. Thank you. No questions. Everything is very, very clear. I really appreciate that. And I appreciate all the different perspectives to coming at this, you know.
Mhm.
So thank you.
Excellent. Well, if for the grant, especially given the mechanism, if you need a letter saying that there's, I don't know how you want to put it, well, however you want to word it. Like, I don't think you want to put a consultant on because that might look weird. But the fact that we have an existing relationship between the Moores with the clinicians and neurologists, and that we work together and we attend seminars together, that kind of thing. We can get those and just figure out what you would like in the letter, and then we can figure out who should write the letter, because that might be Liz or me or Rod
Sure.
Scott or somebody. Just that there's other connections for you in terms of the overall development of the idea.
Yeah. Well, and I think that's going to be really important, given who you're competing against, to show that yes, they'll say we're a small institution, but you're part of this research-rich community of neuroscientists that that can help you with collaborations and interpretations and advice, and because that's what they'll be concerned about.
As part of that, I wonder, like, about inviting Hakeem or anybody in like our sort of broader network to those neuroscience seminars that we do on Wednesdays. Because really we're hoping to like build that community, and you would fit perfectly in it. And like, there's a lot of like neuroscience adjacent people, but like we want to build that community, and it's done virtually too. So it's not hard to do that from anywhere.
They're Wednesdays, right?
It's every other Wednesday at 4.
Okay.
So in person, I think we're still getting pizza. It started a little bit pre-COVID, and then it kind of became all virtual. But it's good, and I think it's a good idea, and start pulling in the DSU folks, starting with Hakeem. Yeah.
Yeah, especially since it's virtual and it doesn't require a 3-hour time commitment.
No, that's right.
For the drive and
that's correct.
Yeah. Yeah, and then you can have like chances to present stuff you're working on, or just present ideas. I did like a grant presentation idea once, and just getting people's feedback was great.
It's good. It's relative. I mean, the group's relatively informal, so it's you can pitch ideas. Slide equivalents of chalk talks. That's what some people do. But it's it's well attended, and there's usually clinician scientists. Rod, the chair for the neuroscience group, attends. It's a good opportunity, and then we can say that we're already doing it. Right? You do it before you submit.
That's great.
So we have not only do we have this, but we're already taking advantage of it. All right, great, nice. It's really exciting work.
Awesome.
Yeah.
Enthusiastic.
If you want anybody to review
thank you so much. I
your Ames page again, I'm happy to look at it when you revise it.
Yeah.
Okay. Okay. I'll take you offline. Thank you.
Okay.
Good luck.
All right.
Yeah.
Thank you all.
Thanks, everybody.
Have a great weekend, everyone.
You too. Thanks, Rob.
Thanks.
Thanks, Melissa.
Nice to meet you, Melissa.
Thanks,
Nice to
Julianne.
meet you
Thanks,
too.
Aaron.
Bye.
Thanks, everyone. Take care.